A UCLA study challenges the traditional understanding of genetic instructions in stem cells, particularly mesenchymal stem cells found in bone marrow. These cells secrete therapeutic proteins, and the study found that the amount of protein released did not strongly correlate with the expression of genes coding for that protein. Instead, the researchers identified other genes, including one coding for a cell surface protein called IL-13RA2, which showed a strong correlation with protein secretion. Cells with IL-13RA2 secreted 30% more of the therapeutic protein, and in a culture experiment, they continued to secrete 60% more protein after 6 days. The findings could impact the efficiency of manufacturing antibody-based treatments and enhance the development of cellular therapies. The study utilized nanovials, microscopic hydrogel containers, to capture single cells and their secretions, allowing the researchers to connect protein secretion to gene expression on the single-cell level. The study opens up new questions and opportunities for advancing biotechnology and regenerative medicine research.
If you enjoyed this video, please give it a thumbs up and subscribe to my channel for more great content.